It is, without a doubt, the absolute most frustrating human struggle. You're just lying there, you're staring at the ceiling, the clock says like 3:00 a.m., and you were just so desperate for sleep.
Oh, yeah, it's awful.
Right, and you're doing that terrible mental math. Like, okay, if I fall asleep right this exact second, I can get exactly 3 hours and 42 minutes of rest, and the harder you try, the more awake you feel.
It's a uniquely agonizing experience, really, because the anxiety about not sleeping, well, it actually becomes the very thing that's keeping you awake. It's this self-perpetuating loop of physiological arousal.
Exactly, and usually the advice you get is basically just this superficial checklist. Drink some chamomile tea, turn off your phone, maybe count some sheep.
Yeah, the standard sleep hygiene stuff.
Right, but today we are going on a deep dive into the hidden architecture of sleep. Because I've been looking at this massive stack of sources we pulled for today's deep dive. And it's everything from a practical medical primer on behavioral therapies for insomnia to this incredibly dense, comprehensive physiological review on how sleep interacts with the immune system.
A lot of material to get through.
It is. And there's also this cutting-edge neurological paper published in Frontiers in Aging Neuroscience. And I gotta say, the overarching theme here is just wild. We aren't just going to give you a list of ways to fall asleep. We're going to explore the mind-blowing biological mechanics of why your body demands sleep in the first place.
Yeah, the science we're looking at fundamentally shifts how we view rest. We're going to examine what actually happens inside your brain and, your immune system when you finally drift off.
Because it's not what people think, right?
Not at all. It's not this passive state of just shutting down. It's actually a highly active, really aggressive period of biological maintenance.
Right. And before we are done here, you're going to learn how simply taking a specific type of breath can literally, physically pump fluid through your brain tissue.
Which is still just incredible to me.
It's crazy. And we'll uncover why the posture you sleep in, like specifically sleeping on your side, might actually be a neurological superpower. Plus, how this bizarre 1980s scientific search for a quote-unquote sleep toxin accidentally revealed a deep, fundamental link between your sleep cycle and your immune system fighting off infections.
There is so much interconnected biology here.
So let's unpack this. Let's start right at the threshold of sleep. So you're in bed, your mind is racing with the day's stress, maybe financial strains, burnout, whatever. How do we actually transition from that high alert state into rest? The sources mentioned that cognitive behavioral therapy for insomnia, or CBTI, is kind of the gold standard for long-term sleep issues.
It is, yeah. CBTI is fantastic because it addresses the root causes. So, changing your sleep schedule, altering your environment, and really breaking those negative cognitive associations with your bed.
Right, but if it's 3:00 a.m., that doesn't really help me in the moment, does it?
Exactly. When it's 3:00 a.m. and you're staring at the ceiling, behavioral therapy won't just miraculously knock you out in that instant. You need to physiologically manipulate your autonomic nervous system.
Yeah.
And the most accessible, immediate tool for that is your breath.
Which is where these breathing techniques come in. The sources outline a variety of them. And what's interesting is that looking through them, they aren't just like 8 random ways to breathe. They seem to cluster into these distinct biological mechanisms.
They absolutely do.
So let's look at the first cluster, which seems focused entirely on chemical manipulation, specifically carbon dioxide, which completely goes against common sense for me. Like the Buteyko method, for example, has you sit in bed, breathe normally for 30 seconds, and then pinch your nose and hold your breath until you feel this absolute desperate need to breathe again.
The air hunger, they call it.
Yeah. But why on earth would I want to build up CO2 when I'm trying to relax? Isn't oxygen the good guy and CO2 is just the toxic waste gas?
Well, so that is the common misconception. And it's exactly why understanding the chemistry is so crucial here. We tend to think of carbon dioxide merely as like an exhaust fume.
Right.
But in human physiology, carbon dioxide is actually a master regulator. There's this thing called the Bohr effect.
The Bohr effect. Okay.
Yeah. And it dictates how oxygen behaves in your bloodstream. So your red blood cells are packed with hemoglobin, which carries the oxygen. But here's the catch. Hemoglobin won't release that oxygen into your tissues unless the environment is sufficiently acidic.
Oh, wait. And what creates that acidity?
Carbon dioxide.
Hold on. So if I'm stressed out and I'm breathing heavily, like maybe I'm hyperventilating in bed without even realizing it, I'm blowing off too much CO2.
Precisely. You're exhaling all of your carbon dioxide.
And without that CO2, The oxygen just stays locked inside my blood cells and never actually gets into my brain or my muscles to relax them.
You've hit the nail on the head. It's called hypocapnia, or low carbon dioxide, and it's incredibly common in chronically stressed individuals. Yeah, so your blood might be, say, 99% saturated with oxygen, but your tissues are literally starving for it because the hemoglobin just won't let it go.
That is wild.
So techniques like the Buteyko method or even box breathing, where you inhale for 4 seconds, hold for 4, exhale for 4, and then hold empty for 4, they're designed to intentionally build up your CO2 tolerance.
Okay, so by normalizing the carbon dioxide levels, I'm basically dilating my blood vessels and letting that oxygen finally release into my starving, stressed out tissues.
Exactly. It's about utilizing the air better, not just getting more of it.
That totally reframes everything. And that same principle seems to apply to the 4-7-8 technique, the one developed by Dr. Andrew Weil.
Right, based on ancient pranayama.
Yeah, where you inhale for 4 seconds, hold for 7, and then exhale with this loud whooshing sound for 8 seconds. So the 7-second hold builds the CO2, but what is the incredibly long 8-second exhale actually doing?
So the prolonged exhalation is a mechanical signal to your brain's threat detection center. Think about it evolutionarily. If you were sprinting away from a predator, you would be taking rapid, shallow breaths.
Pufferfish breathing, basically.
Right. You couldn't possibly sustain an 8-second agonizingly slow exhalation if you were running for your life.
Oh, I see.
So by consciously forcing your body to exhale twice as long as you inhale, you are sending a retrograde signal through your nervous system. You're basically telling your brain, hey, I am safe, the threat is gone.
You're faking it till you make it biologically.
Basically, it forcefully shifts you from sympathetic dominance, which is your fight or flight, into parasympathetic dominance, which is rest and digest.
Okay, so that's the chemical side and the evolutionary signaling. But then there's this second cluster of techniques in our sources that seem almost entirely mechanical.
The physical manipulation of the breathing apparatus.
Exactly. Like the Papworth method, or diaphragmatic breathing. You put one hand on your chest and one on your stomach, and you force only the stomach to rise. And the source mentions this is particularly effective for people who habitually sigh or yawn. What's the link there?
Well, habitual sighing is a classic clinical indicator of a dysregulated autonomic nervous system. It's a sign of chronic shallow breathing.
Oh, really? I sigh all the time when I'm stressed.
A lot of people do. When we stress breathe, we use these secondary muscles of respiration up in our chest and our neck. It's highly inefficient and it's exhausting for the body.
So we need to use the diaphragm instead.
Yes. Engaging the diaphragm, which is that massive parachute-shaped muscle at the base of your lungs, it does a few critical things. First, it pulls air deep into the lower lobes of the lungs where blood flow is actually the richest, so you get better gas exchange.
Okay, makes sense.
But more importantly for sleep, the actual physical descent of the diaphragm into your gut massages your abdominal organs and physically stimulates the vagus nerve.
The vagus nerve. That comes up so much in health optimization circles. It's essentially the superhighway of that parasympathetic rest and digest system, right?
It absolutely is. It is the primary conduit of calming signals from the body up to the brain.
And that actually brings us to what might be the most fascinating breathing technique in these sources. Because there's one that targets the vagus nerve directly through physical vibration, the Brahmari pranayama.
The humming breath, it's a very unique one.
It is highly sensory. Like you close your eyes, take a deep breath in, cover your ears with your hands, and place your fingers gently over your eyes and the sides of your nose. And as you exhale slowly, you make this humming sound.
Did you try it?
I actually did. I tried it earlier today when I was reading through the source material, and it feels completely bizarre at first. But the resonance in your skull is undeniable. The source points to research showing this rapidly drops your heart rate. But how does just humming actually change my cardiac rhythm?
Well, it all comes back to the anatomy of that vagus nerve. So branches of the vagus nerve run right near the vocal cords, and they also run up through the face and the ears.
Oh, so you're physically touching the nerve pathways.
Exactly. The physical vibration of the hum, combined with that gentle acupressure on the face and the extended exhalation, it provides this massive multisensory stimulation to the vagus nerve.
That's incredible.
You are literally vibrating your nervous system into a state of calm. You're overriding those anxious cognitive loops that are keeping you awake by just flooding the sensory channels with soothing mechanical feedback.
Okay, so I'm doing the diaphragmatic breathing, I'm building my CO2 tolerance, I'm stimulating my vagus nerve, my chest is expanding and contracting deeply like a giant bellows. And this is where the research takes a massive, totally unexpected turn.
It's my favorite part of the data.
because our sources reveal that those deep pressure changes in the chest don't just calm you down, they actually act as a mechanical pump for a hidden plumbing system deep inside your brain.
Yeah, the transition from the breath to this internal plumbing system is, I think, one of the most exciting frontiers in modern neuroscience right now.
It's like finding a secret room in a house you've lived in your whole life.
That's a great way to put it, because once that breath helps you cross the threshold into actual sleep, your brain initiates this biological process that scientists literally didn't even know existed until 2012.
Let's talk about the history of this, because
Yeah.
it's wild that we missed something so fundamental. I mean, the rest of the human body has a lymphatic system. We all know that. We have this network of vessels running parallel to our blood vessels that clears away cellular waste, large proteins, just the general trash of daily cellular life.
Right, it filters through the lymph nodes.
Exactly, which is why your neck gets swollen when you're sick.
Hmm.
But for over a century, the scientific consensus was that the brain, the most metabolically active organ in the entire body, the one producing the most trash, just didn't have one.
It was considered a massive anatomical paradox. Seriously, how could an organ that consumes 20% of the body's energy survive without a dedicated waste clearance system?
It makes no sense. What did they think was happening?
Well, the old assumption was that brain cells just slowly recycled their own waste, or that the waste kind of diffused passively into the cerebrospinal fluid in this very slow, disorganized way.
But that wasn't enough.
No, not even close. Passive diffusion isn't nearly fast enough to clear the sheer volume of metabolites that the brain produces every day.
Right. Then in 2012, researchers led by Dr. Macon Niedergard at the University of Rochester discovered something revolutionary. They found the hidden plumbing, and they called it the glymphatic system.
Yeah, it's a mashup word. Glial cells plus lymphatic function. Glymphatic.
Okay, so before we can understand how this washes the brain, we need to map out the terrain a bit, because the sources outline a very complex fluid dynamic inside the skull. I want to make sure I had this straight, so please correct me if I'm wrong here.
Go for it.
So let's say the brain is basically a city, and in this city, you have different types of water. You have the blood, which is running in the main, sealed pipes. You have the interstitial fluid, or ISF, which is the water actually out on the streets touching the buildings, the brain cells.
Right.
And then you have cerebrospinal fluid, or CSF, which is this specialized clear fluid made deep in the brain that acts like a protective cushion.
That's a really strong foundational analogy. And the key here is that separating these different fluids are highly selective borders.
Like the blood-brain barrier.
Exactly. The most famous is the blood-brain barrier, the BBB. Its job is to keep the blood tightly separated from the interstitial fluid on the streets. You absolutely do not want blood physically touching your neurons. It's highly toxic to them.
Oh, wow. Good to know.
But you also have the blood-cerebrospinal fluid barrier, the BCSFB, and that controls the filtration of blood to create that clear CSF in the first place, primarily in a specialized structure called the choroid plexus.
Okay, so the choroid plexus manufactures this super clean CSF water, and then it flows out into the subarachnoid space, which is, from what I understand, this fluid-filled area wrapping around the entire outside of the brain.
Yep, the surface level.
But how does this clean fluid actually get deep down into the city to wash the streets? The sources describe this incredibly specific, almost architectural journey. Arteries run along the surface of the brain, and eventually they have to dive straight down into the dense brain tissue to deliver oxygen.
Right, they branch out and penetrate the tissue.
And when they dive down, they become penetrating arteries. And this is where the magical structure is formed.
Yes. As those arteries plunge down into the brain tissue, they don't just push through the cells nakedly. They actually bring a little sleeve of that subarachnoid space down with them.
A sleeve.
Yeah, it creates what is known as the perivascular space. Historically, these were called Virchow-Robin spaces.
Let me try to visualize this for you. Imagine the blood vessel is a solid pipe, and surrounding that pipe, there is a microscopic donut-shaped tunnel. That tunnel is the perivascular space.
Perfect visualization.
So the inner wall of this tunnel is the blood vessel itself. But what is the outer wall made of?
The outer wall is entirely constructed by the brain's support cells, specifically a type of glial cell called an astrocyte.
I want to pause on astrocytes for a second because they sound incredible. They are shaped like stars, right?
Yes, astro meaning star. Astrocytes are essentially the nursemaids of the brain. They actually outnumber neurons, they provide nutrients, they regulate electrical signals, and as we now know from this research, they build the plumbing.
How do they build it?
Well, they have these long physical projections called end feet. The astrocytes wrap their end feet completely around the perivascular space, tiling together to form a solid, continuous outer tube.
Like bricks in a sewer wall.
Exactly. And embedded in these astrocytic end feet are millions of highly specialized water channels. They're called aquaporin 4, or AQP4 channels.
Okay, so I'm picturing it. The clean CSF is traveling down from the surface, along the outside of the artery, inside this donut-shaped perivascular tunnel. But it needs to get out of the tunnel and onto the city streets to actually wash the brain cells.
It has to cross the barrier.
And it's driven by two main pumps, right? One is the physical pulsing of the artery itself. So every single heartbeat expands the artery and pushes the fluid down the tunnel. And the second pump is our deep breathing from those respiratory techniques, creating pressure waves in the chest that travel up the blood vessels.
That's the mechanical link, yes.
So this pressure forces the CSF through those aquaporin 4 water channels on the astrocyte end feet, and boom, it sprays out onto the streets.
Exactly. The clean CSF crosses into the interstitial space, and once it's there, it becomes a pressurized wave sweeping through the dense forest of brain cells, mixing with the dirty interstitial fluid.
And taking out the trash.
It mechanically flushes out the metabolic waste products, the rogue proteins, and the cellular debris that accumulated during your waking hours.
And once it's collected all that trash, it has to drain somewhere. It can't just sit there. So it sweeps through the tissue toward the veins. It enters another donut-shaped tunnel around the veins. The perivenous space flows back out to the surface and ultimately exits the skull through meningeal lymphatic vessels,
which drain right down into the lymph nodes in your neck.
The city is officially washed. But there is a detail in the source that completely blew my mind. The researchers found that this sweeping wave of fluid, this whole washing process, is almost non-existent when you are awake. It only happens during sleep. Why is that?
This is where we uncover the biological prerequisite for the wash. The researchers discovered that during sleep, the actual physical space between the brain cells, the interstitial space, it expands by up to 60%.
Wait. The cells physically shrink.
They do. They literally change size.
That is insane.
It's amazing. In the waking state, your brain is constantly bathed in noradrenaline, which is a neurotransmitter associated with arousal and alertness.
Right. Keeps you focused.
And noradrenaline acts on the brain cells, causing them to physically expand and plump up. If we go back to your city analogy, imagine the actual buildings suddenly inflating.
Oh, wow. The streets have become incredibly narrow and congested.
Exactly. The physical resistance to fluid flow becomes so high that the glymphatic fluid just bounces off. It can't penetrate the tissue. It just flows along the surface.
So awake means high noradrenaline, plump cells, narrow streets, no wash. As soon as we fall asleep, the noradrenaline shuts off, the cells shrink back down, the streets open up, and the floodwaters rush in.
That dramatic shift in cellular volume is the switch. It's an elegant, deeply mechanical system.
It really is.
But understanding how perfectly it's designed also reveals how catastrophically it can fail. The implications of the system breaking down are fundamentally shifting our entire understanding of neurodegenerative diseases.
Yeah, let's go there. Because if a single physical blow, or just bad habits, can misalign this system and ruin our brain's plumbing, it begs the question, what happens when the trash doesn't get taken out?
Nothing good.
The sources highlight two specific pieces of neural trash that the glymphatic system is responsible for clearing: amyloid beta and tau proteins. And anyone who has had a loved one with Alzheimer's disease knows those exact names.
They're notorious.
They're the toxic proteins that clump together to form plaques and tangles, destroying memory and cognition.
Right, and for decades, Alzheimer's research focused heavily on the overproduction of these proteins. Or they tried to find drugs to break down the plaques once they had already formed.
Which hasn't been super successful.
Not very. But the glymphatic research provides a completely new paradigm. It gives us a mechanism for why they build up in the first place. In Alzheimer's models, researchers observed this phenomenon called AQP4 depolarization.
Let's break down that word, depolarization.
Yeah.
Because normally those aquaporin 4 water channels are highly concentrated right at the tips of the astrocyte end feet, lined up perfectly with the perivascular tunnel to pull the water through. What happens when they depolarize?
It's essentially a cellular distress response. When astrocytes are subjected to chronic inflammation or injury, they become what we call reactive.
Okay.
And part of this reactivity involves changing their physical cell membrane structure. The AQP4 channels lose their targeted anchor points at the end feet, and they just sort of spread out over the entire surface of the astrocyte.
So the water channels are no longer lined up with the plumbing. It's like moving the drain from the bottom of the sink to the side of the wall.
That's a great analogy.
The fluid flow just stalls out.
The flow stalls, and the clearance of amyloid beta drops precipitously. And the most startling finding in the research is that this astrocytic depolarization, this glymphatic malfunction, it actually precedes the significant deposits of amyloid plaques.
Wait, so the plumbing breaks before the trash piles up?
Yes. It strongly suggests that a failure of the brain's macroscopic plumbing system isn't just a symptom of Alzheimer's. It might be the foundational trigger.
Wow. And the sources show this mechanism holds true across multiple horrific diseases. Take Parkinson's disease, for example. Parkinson's involves the accumulation of a different toxic protein called alpha-synuclein, which destroys the dopamine-producing neurons involved in movement. How did researchers test the plumbing connection here?
They used Parkinson's mouse models and performed a rather dramatic intervention. They surgically ligated or physically blocked the meningeal lymphatic vessels.
They literally plugged the final exit drain in the neck.
Exactly. They stopped the outflow.
And what happened?
The results were severe and rapid. The alpha-synuclein proteins backed up into the brain tissue immediately, triggering massive local inflammation. The astrocytic plumbing broke down even further, and they observed accelerated death of those critical dopaminergic neurons.
So a mechanical blockage downstream caused a neurodegenerative cascade upstream.
Precisely.
Which perfectly explains the data on traumatic brain injury, or TBI. This is a terrifying section of the research for anyone involved in contact sports or anyone who's had a bad fall.
That's sobering data.
When someone takes a hard physical blow to the head, the kinetic energy shears the brain tissue. And the trauma physically damages the astrocytes, causing them to lose that AQP4 polarity. Studies show that after a TBI, glymphatic fluid flow drops by a staggering 60%.
Just plummets.
But the truly scary part isn't the drop itself, it's how long it lasts. The sources say it stays suppressed for over a month after a single injury.
A month of impaired clearance. Think about that. That is 30 days where tau proteins are accumulating at an accelerated rate, laying the groundwork for chronic traumatic encephalopathy, or CTE.
The football player disease.
Right. It recasts the concussion from what we thought was just a temporary electrical disruption into a long-term structural mechanical plumbing failure.
And it isn't just violent trauma that does this. The sources highlight that diabetes wreaks havoc on this system too. How does high blood sugar affect the brain's water channels?
So in type 2 diabetic models, researchers used MRI contrast agents to actually track the fluid flow, and they found the glymphatic clearance rate was 3 times slower compared to healthy models.
3 times slower.
Yeah. The chronic high blood sugar and the systemic metabolic dysfunction, it damages the blood vessels over time. This impairs the arterial pulsations, which, remember, is the main pump for the fluid.
Right, the heartbeat pump.
Exactly. It also causes the perivascular space to abnormally expand, which disrupts that tight astrocytic wrapping, so wastes accumulate, triggering local inflammation, which damages the vessels even further.
Wow.
It's a vicious, accelerating cycle of retention and cognitive decline.
It's a terrifying array of diseases, and they all link back to this one system. Alzheimer's, Parkinson's, CTE, diabetic cognitive decline. So if this brainwashing system is our primary defense against losing our minds as we age, the absolute most pressing question for you listening right now is, how can I optimize my own glymphatic system tonight?
What are the actionable steps?
Exactly. What are we doing to this plumbing system every single day through our normal habits? What happens when we drink or exercise or just, change how we lie in bed?
The lifestyle livers are remarkably potent, and the science behind them is fascinating. Let's start with the most basic mechanical liver. Your posture in bed.
Yes. The sources outlined studies that tested fluid transport across different sleeping positions. They looked at supine, which is on your back, prone on your stomach, and lateral on your side. And they found that sleeping on your side is scientifically proven to be the most efficient posture for glymphatic transport and for clearing out toxic amyloid beta.
It's incredibly consistent data.
I have to admit, when I read this, it felt a little absurd. You're telling me that simply rolling over onto my side is a biological defense mechanism against dementia? Like, how can gravity possibly have that much of an effect on microscopic brain channels?
It seems overly simplistic, I know, but it makes profound anatomical sense when you look at the cardiovascular system as a whole.
Okay, walk me through it.
So, the driving force pushing the fluid into the brain is the pulsation of the major arteries in the neck. The efficiency of that pulsation is entirely dependent on the pressure gradients in your chest and neck. When you sleep on your side, the lateral position, you optimize venous return to the heart.
meaning the blood gets back to the heart easier.
Right. It prevents the pooling of blood, and it normalizes the pressure in your jugular veins. And your jugulars are the main outflow tracts from the brain.
Ah,
If the jugular pressure is too high, the glymphatic fluid literally can't drain out of the skull. Furthermore, lateral sleeping optimizes sympathetic nerve activity and your respiratory mechanics. You are basically aligning the macroscopic pumps of your body to push the microscopic fluid in your brain.
so you're basically just straightening the kink in the hose.
That's exactly it.
Let's talk about a chemical lever,
Yeah.
alcohol. Because the data here is incredibly nuanced. The sources say it is a double-edged sword. If you consume high doses, like binge drinking, it essentially stops the brainwash dead in its tracks. What's the mechanism there?
High doses of alcohol induce massive acute inflammation in the brain tissue. It's toxic. The astrocytes detect this toxicity and they become highly reactive.
There's that reactivity again.
Yep. The research points to a huge surge in the expression of a protein called GFAP, which is a key marker of astrocyte stress. And as we discussed earlier, stressed astrocytes depolarize.
They move the drain.
Right. They retract their end feet, mislocate the aquaporin 4 channels, and shut down the perivascular pathways. Binge drinking literally shuts the storm drains in your brain.
But the nuance you mentioned is the wild part to me. The same studies found that low doses of alcohol, meaning chronic low-level exposure, like maybe a single glass of wine with dinner, actually promoted waste clearance. How can a toxin be helpful?
It comes down to vasodilation. Low doses of alcohol act as a mild vasodilator, meaning they relax and widen the blood vessels.
Okay.
This widening may promote a really healthy interaction between the endothelial cells lining the blood vessel and the smooth muscle cells surrounding it, which generates nitric oxide.
And nitric oxide is good for flow.
Exactly. This relaxed, widened state of the vasculature actually eases the diffusion of metabolic wastes from the interstitial space into the perivascular drainage tunnels.
That's a very tight rope to walk, though. A little bit widens the pipes, a little bit more stresses the cells and shuts the whole thing down.
Very tight. It's all about dosage.
What about exercise? We all know working out is good for cardiovascular health, but the sources indicate it acts directly on the glymphatic channels, too.
Yes, it does. In preclinical trials where mice were given access to voluntary wheel running, researchers didn't just see faster clearance rates, they actually saw structural improvements.
Structural, like the brain changed shape.
The plumbing did. Regular exercise actively maintains and improves the targeted polarization of those AQP4 channels. It physically reinforces the structural integrity of the brain's plumbing over time.
That's amazing. Now, you mentioned preclinical trials with mice. The sources dedicate a significant amount of text to how these researchers actually see this invisible fluid moving in a living brain. And the technology is mind-blowing. They use something called 2-photon fluorescence imaging. Can you break down how scientists actually observe this microscopic process?
Oh, it's a total marvel of modern biomedical engineering because traditional microscopes, they can't see deep into living tissue. The light scatters too much.
Right, it's opaque.
So 2-photon microscopy uses highly focused, ultra-fast pulses of infrared lasers, and infrared light penetrates much deeper into the tissue without damaging it. So what they do is researchers inject fluorescent tracer dyes directly into the cisterna magna, which is a pool of cerebrospinal fluid right at the base of the mouse's skull.
Okay, so they dye the fluid.
Yes. And then, using these lasers, they can watch in real time in a living, breathing, sleeping mouse exactly where that glowing fluid travels, how fast it moves, and exactly where it gets stuck.
And they used this exact technology to study different states of unconsciousness, specifically anesthesia, because going under the knife is obviously a deep state of unconsciousness. But the researchers found that not all anesthetics are created equal when it comes to the brainwash, did they?
They absolutely did not. They discovered that the glymphatic system is exquisitely sensitive to the specific neurochemical cocktail in the brain, not just the mere absence of wakefulness.
Being unconscious isn't enough.
Exactly. When they anesthetized mice with a specific mixture of ketamine and xylazine, the 2-photon imaging showed this beautiful high-flow state that perfectly mimicked natural sleep. The fluid washed through vigorously.
Okay.
But when they used a different, very common anesthetic called isoflurane, the flow was actively inhibited. It stalled.
Meaning if you are under isoflurane, you are unconscious, sure, but your brain isn't cleaning itself at all.
Precisely. It highlights that natural sleep provides a very specific, evolutionarily honed neurochemical environment. You need that low noradrenaline, you need specific brainwave frequencies. Simply being knocked out doesn't necessarily replicate the biological triggers for the wash.
Wow. Okay, so we have firmly established the magnificent mechanics of how sleep washes the brain. The cells shrink, the streets expand, the fluid rushes in, and the toxic proteins are cleared out to stave off dementia.
The nightly rinse cycle.
But what about the rest of the body? Let's leave the skull for a moment and look at the entire organism. What is your immune system doing while you are lying there, paralyzed in the dark?
Moving from the fluid dynamics of the brain to the cellular dynamics of the immune system reveals a completely different, but honestly equally profound evolutionary purpose for sleep.
Yeah.
We are looking at a bidirectional highway where sleep and immunity are inextricably linked.
The history of how we discovered this link is so fascinating and honestly a little embarrassing for early science.
It took us a while to get there.
It really did. The sources start way back in 350 BC. Aristotle wrote a whole treatise called On Sleep and Sleeplessness, and he observed that when people eat a heavy meal, they get sleepy. He also noticed that when people get sick with a fever, they get incredibly sleepy.
A very astute observation.
Right. So he concluded that sleep was caused by, quote, hot vapors rising from the stomach during digestion or illness, physically choking off the brain.
I mean, it was a logical deduction based on observation at the time, but obviously mechanically flawed.
Just a bit. So fast forward to the early 20th century, and scientists had moved past vapors. They began searching for a physical substance in the body. The leading theory was the hypnotoxin theory.
Ah, yes, René Pierron.
Yeah, this French scientist, René Pierron, theorized that while we are awake, our bodies slowly accumulate a toxic chemical in our blood. And when this hypnotoxin reaches a critical threshold, it literally paralyzes the brain, forcing us to sleep so the body can clear it out.
So for decades, scientists were desperately searching for this human chemical. In the 1960s, a Harvard researcher named John Pappenheimer did these grueling experiments. He extracted cerebrospinal fluid from sleep-deprived goats and actually injected it into rats.
Oh, wow.
And the rats immediately passed out, so he knew the substance was in there somewhere.
You just couldn't isolate it.
Right. Finally, in the 1980s, technology caught up, and scientists successfully isolated the very first known sleep-promoting factor. But the discovery sent absolute shock waves through the scientific community.
Because it wasn't a human molecule at all.
The revelation was utterly paradigm-shifting. The substance they isolated was neurodegeneration.
Wait, hold on. Let me get this straight. You're telling me scientists spent a century looking for the human chemical that causes sleep, and they ended up finding literal bacteria?
Yes.
Because neurodegeneration is a structural component of bacterial cell walls. How is that even possible? Are we infected every time we get tired?
It sounds like science fiction, I know. But it was the key that unlocked the bidirectional link. It wasn't an infection. It was proof of a shared evolutionary language. What do you mean? When researchers introduced these microbial fragments, these neuropeptides, into animal models, the animal's immune system detected them. And in response, the immune system deployed its own signaling molecules, which then traveled up to the brain and induced profound, deep sleep.
Oh my God.
The hypnotoxin wasn't a sleep chemical at all. It was an immune trigger.
Let's walk through the mechanics of that first direction.
Yeah.
Infection causing sleep. So let's say I catch a nasty flu virus. A virus enters my respiratory tract. My innate immune system has these highly specialized, really ancient sensors called pattern recognition receptors, or PRRs.
Right.
They're constantly scanning the environment, acting like molecular border patrol.
And what they are scanning for are pathogen-associated molecular patterns, or PAMPs. These are structural signatures that simply don't belong in the human body. Like the double-stranded RNA of a virus, or the lipopolysaccharides of a bacterium. When a pattern recognition receptor locks onto a PAMP, the alarms sound.
And the alarms are essentially a massive inflammatory response. My immune cells start rapidly pumping out chemical messengers called cytokines. The sources specifically highlight tumor necrosis factor, or TNF, and interleukin-1 beta.
IL-1 beta, yes.
Right. And these are the chemicals responsible for the absolute misery of being sick, right? The fever, the body aches, the systemic inflammation.
Yes, they mediate sickness behavior.
But crucially, the research shows these exact same cytokines leave the infection site, travel through the bloodstream, cross into the brain, and actively command the sleep centers to alter my brain waves.
Specifically, TNF and IL-1 beta act on the hypothalamus and the basal forebrain to dramatically increase both the duration and the electrical intensity of slow-wave sleep.
So when I get the flu and I feel like I've been hit by a freight train and I end up crawling into bed and sleeping for 14 hours straight, My immune system is literally drugging my brain with cytokines to paralyze me in deep sleep.
That is precisely the mechanism. In biology, we literally call it sickness behavior. We tend to think of lethargy and sleepiness just as like unfortunate side effects of feeling unwell.
Just feeling crummy.
Right, but it is actually a highly calculated, aggressive evolutionary survival strategy.
Really?
Yes. Your body is facing an existential threat. Mounting a massive immune response requires an astonishing amount of metabolic energy. So the immune system releases cytokines to force you to stop moving, stop foraging, stop expending energy on muscles or digestion.
It grounds you.
It completely shuts down the host so it can redirect every available ounce of energy into raising your core body temperature to burn out the pathogen and arming the adaptive immune system.
And the consequences of bypassing this forced shutdown are terrifying. The sources detail the dark history of sleep deprivation experiments. You literally cannot survive without this shutdown.
No, you can't.
Back in the late 19th century, scientists kept dogs completely awake by constantly walking them, and the dogs inevitably died within a week or two. But the mechanism of why they died wasn't actually understood until the 1980s.
Right, Alan Rechtschaffen's work.
Yeah, this researcher, Alan Rechtschaffen at the University of Chicago, developed this brutal apparatus called the disc over water method to test total sleep deprivation on rats.
It remains one of the most revealing, if, harrowing experiments in sleep science.
Can you describe it?
So Rechtschaffen housed an experimental rat and a control rat on a divided circular disc. And this disc was suspended over a pan of shallow water. Both rats had EEG electrodes surgically implanted in their skulls to monitor their brainwaves continuously.
And the setup was entirely automated.
Yes. The computer monitored the experimental rat's brainwaves. The very second the computer detected the electrical signature of sleep onset, it would activate a motor, and the disc would suddenly rotate.
Oh, wow.
So the experimental rat would be pushed toward the water and forced to walk to avoid falling in, which woke it up instantly.
And what about the control rat?
The control rat was on the exact same disc, so it also had to walk whenever the disc moved. But the key is the control rat could sleep whenever the experimental rat happened to be awake.
I see. So the environment was identical, the physical exercise was identical, the stress of the water was identical, but only the experimental rat was completely, totally deprived of sleep.
Exactly. And the physiological unraveling of the sleep-deprived rats was catastrophic.
What happened to them?
Within a few days, their fur lost its shine and became yellowed and totally disheveled. They developed severe skin lesions all over their bodies. Their core body temperature plummeted, yet their metabolic rate skyrocketed.
They were burning up from the inside.
Basically, they were eating twice as much food but losing weight rapidly. Their endocrine systems just went into massive overdrive.
And how long did they last in that state?
They universally died within 2 to 3 weeks.
But the autopsy results are what truly matter here, based on our sources. Because they didn't die of a heart attack from stress or brain failure from exhaustion. What actually killed them?
They died of massive systemic bacterial infections, septicemia.
Sepsis.
Yes. Even though they were housed in a meticulously clean, pathogen-free laboratory environment, their biological defense mechanisms completely collapsed.
Wait, where did the bacteria come from if the lab was perfectly clean?
They came from inside them.
Oh, gross.
Your gastrointestinal tract is filled with trillions of bacteria that normally live there completely harmlessly, right? They're kept in check by the intestinal mucosal barrier and your active immune system.
Right, our microbiome.
Well, the profound sleep deprivation caused a total failure of host defense. The immune system basically ceased to function, the intestinal barrier broke down, and their own gut bacteria escaped into their bloodstream, causing lethal sepsis.
So no sleep equals total immune collapse. You literally get eaten by your own gut bacteria.
In extreme deprivation, yes, that's what happens.
That paints such a stark picture of sleep as an active shield. But how exactly does sleep arm the immune system to prevent this? Because we need to look closely at the different stages of sleep to understand this, like nightly military training exercise.
Yeah, to understand the immune training, we really have to look at the architecture of the night. When subjects are studied in a sleep lab, researchers use a polysomnogram. They measure brain waves with an EEG, eye movements with an EOG, and muscle activity with an EMG.
The full workup.
Right. And through these tools, we see that sleep is highly structured into distinct phases.
Okay, so you have non-REM sleep, or NREM,
Yeah.
and REM sleep. NREM is broken into 3 stages. You've got N1, which is that really light transitionary sleep. Then N2, which features these rapid bursts of brain activity called sleep spindles and deep draughts called K-complexes.
Yes.
And then N3, which is the crucial slow-wave sleep. During N3, your brain waves slow down to less than 4 hertz, just these huge synchronous rolling waves of electrical activity. And this slow-wave sleep dominates the first half of your night, mostly.
It does, and contrasting that is REM sleep, rapid eye movement. This dominates the second half of the night, closer to morning. During REM, your brain becomes incredibly electrically active, almost indistinguishable from wakefulness on an EEG. You dream vividly.
Right.
And to prevent you from physically acting out those dreams and hurting yourself, your brain stem actively paralyzes your voluntary muscles in a state called muscle atonia.
So what is the immune system doing during all these massive electrical shifts? The sources break down the immune system into 2 main arms. You have the innate immune system, which is your fast, unspecific first responder.
Blunt instrument.
Yeah, it's made of cells like macrophages and neutrophils that just blindly swallow any invader they bump into. But the true heavy artillery is the adaptive immune system. This is slower, highly specific, and made of T cells and B cells.
And the defining characteristic of the adaptive immune system is its ability to form immunological memory.
Meaning it remembers the bad guys.
Exactly. Once it fights off a specific virus, it remembers exactly what it looks like, so it can destroy it instantly the next time it enters the body. And sleep is the crucial biological laboratory where this memory consolidation takes place.
Let's trace the exact mechanism of this memory formation, because it's like a tiny spy movie. Let's say you get exposed to a new virus.
Mm-hmm.
An innate immune cell, specifically an antigen-presenting cell like a dendritic cell, chews up the virus.
Right.
It takes a tiny microscopic peptide fragment of that virus and displays it on its outer surface. Now, this dendritic cell has to travel to a lymph node, find a T cell, and physically bind to it to show it the viral fragment. It's essentially handing over a biological mugshot so the T cell can mount a targeted attack.
That's a perfect analogy. The physical binding between the antigen-presenting cell and the T cell is known as an immunological synapse.
That's the synapse, okay.
And forming a strong, lasting synapse is actually very difficult. This is where sleep becomes absolutely vital. The sources show that during early, slow-wave sleep, the body undergoes a massive hormonal shift.
What kind of shift?
Levels of cortisol, adrenaline, and noradrenaline, the stress hormones plummet to their lowest levels of the entire 24-hour cycle. Simultaneously, levels of growth hormone and prolactin surge upwards.
But why does that specific hormonal cocktail matter for those 2 cells trying to talk to each other?
Because stress hormones like adrenaline and cortisol actively interfere with the ability of immune cells to bind together. They literally suppress immune adhesion.
Oh.
Yeah. So by dropping those stress hormones to near zero and elevating prolactin, slow-wave sleep provides the perfect, undisturbed chemical environment for the dendritic cell to lock onto the T cell. The adhesion is strengthened. The information is passed efficiently.
It's as if the immune system needs absolute quiet to have a highly technical conversation. It sounds exactly like your body is sitting at a computer terminal, uploading the latest virus definitions to a central database.
That is an incredibly apt way to conceptualize it. Sleep is teaching your T cells and B cells what to look for, facilitating clonal expansion where the T cells multiply, and ensuring your body is faster and more efficient at fighting off that specific bug the next time.
This mechanism has massive, undeniable, real-world implications for how we live. Like, what happens if you live in a state of chronic sleep deficiency? What if you are a shift worker or a stressed parent or just someone pushing the limits, and you're only getting, say, 5 hours a night for years?
The clinical data on chronic sleep restriction is stark. If you don't achieve sufficient slow-wave sleep, your T cells remain sluggish. The adherence between the cells fails. The immunological synapse simply isn't maintained long enough to form a strong memory.
And the sources cite studies involving vaccinations that prove this undeniably. Researchers gave healthy human subjects a standard vaccine, like a hepatitis A or a flu shot.
A controlled immune challenge.
Right. They let half the group sleep normally, and they severely restricted the sleep of the other half for just a few days following the shot. When they measured the blood weeks later, the sleep-deprived group produced less than half the amount of specific antibodies compared to the rested group.
Less than half. The vaccine was exactly the same. The dose was the same. The immune system was simply robbed of the biological time it needed to process the information and build the weapon.
That's terrifying.
It is. Your immune response is demonstrably physically weaker without sleep.
And the cascade of failure continues. Chronic sleep restriction doesn't just make you vulnerable to viruses. It turns your immune system against your own body. because the immune system isn't getting its nightly reset time. It stays in a mild, continuous state of panic.
It's always on edge.
Right, so it constantly pumps out low levels of inflammatory cytokines, resulting in systemic, low-grade inflammation.
And this chronic inflammation is really the unifying thread that ties everything we've discussed today together. Well, this low-grade inflammatory fire damages the endothelial cells lining your blood vessels all over your body. It contributes to atherosclerosis and systemic vascular resistance.
Which circles right back to the beginning of our deep dive. Because if your blood vessels are inflamed and stiffened, they lose their elasticity.
Exactly.
That impairs the strong arterial pulsations. And without those strong arterial pulsations, the glymphatic system loses its mechanical pump.
Yes.
The perivascular spaces stall, the toxic amyloid beta builds up, the astrocytes depolarize, and you accelerate neurodegeneration.
It is all one massive, intricately interconnected web of biology. A failure in sleep architecture causes an immune failure, which causes vascular damage, which causes a failure in brain plumbing, which ultimately leads to cognitive decline.
Man, we have covered incredible ground today. We started with the simple, frustrating act of just lying awake in bed. We explored how taking a slow, intentional breath, building that CO2 tolerance, and mechanically dropping the diaphragm can stimulate the vagus nerve and initiate the parasympathetic cascade.
The physical levers.
Right. And then we realized that the pressure changes in our chest actually act as a mechanical pump for a newly discovered nightly brainwashing cycle. The glymphatic system literally relies on our brain cells shrinking by 60% so that fluid can rush in and physically sweep away the toxic proteins that cause Alzheimer's, Parkinson's, and CTE.
And from there, we expanded to the whole organism, discovering that this period of unconsciousness is anything but passive.
It's a war room.
It is a highly active biological laboratory. It is the time when your immune system leverages profound drops in cortisol to communicate, to form immunological memories, and to literally arm your T cells to keep you alive in a pathogen-filled world.
Understanding this machinery gives you such a profound sense of empowerment, I think. The next time you are lying in bed, you aren't just trying to turn off or escape the day. Whether you are specifically choosing to sleep on your side to optimize your jugular venous return and maximize your amyloid beta clearance, or you're doing diaphragmatic breathing to kick your nervous system into gear.
Or fiercely protecting your 8 hours before getting a flu shot to boost your antibody production.
Yes, you now know the deeply mechanical why behind the what. You aren't just resting, you are actively performing cellular maintenance.
And viewing sleep through this lens of active maintenance leaves us with a truly provocative thought for the future of medicine.
I love where this is going.
If we now know through rigorous preclinical imaging and physiological data that neurodegenerative diseases like Alzheimer's might fundamentally be plumbing issues caused by poor fluid dynamics,
Right.
and if catastrophic immune failures and systemic inflammation are directly linked to poor sleep architecture,
Yes, and.
could the future of treating these devastating diseases, Alzheimer's, Parkinson's, chronic inflammatory disorders, rely less on finding exotic new chemical drugs to break down plaques after the damage is already done, and more on finding technological ways to artificially induce, enhance, and perfectly manipulate deep sleep? What if we could design a device that perfectly aligns the arterial pulsations with the slow-wave brain frequencies to maximize the glymphatic wash? What if the ultimate, most potent medicine in our future is simply a perfectly engineered night of rest?
That is a staggering way to look at the future of healthcare. So the next time you find yourself staring at that ceiling at 3:00 a.m., doing the mental math of how many hours you have left, don't panic. The anxiety will only spike your noradrenaline and close the channels. Just take a deep, slow breath in through your nose, hold it, let the chemistry shift. And as you let it out on a long, slow exhale, imagine the fluid rushing through your brain, washing the streets clean, while your immune system quietly uploads the code to keep you safe.